There is a question that almost no one asks, and it has been haunting the edges of biology for 20 years. It is not whether life exists elsewhere in the universe. It is whether a second, entirely separate form of life exists here, on Earth, right now. Not bacteria you haven't heard of, not a new species in the Amazon. Something built from fundamentally different chemistry, invisible to every test we currently use to detect living things. Something that has been coexisting with us, perhaps for billions of years, without our knowing. When scientists search for alien life, they build instruments tuned to the signatures of life as we know it. They look for our DNA, our proteins, our particular flavor of carbon chemistry. But if life arose on Earth a second time, independently, from different starting materials, following different rules, we would not see it. Our instruments would pass right through it. Our sequencers would not register it. Our cultures would not grow it. It would be invisible, not because it is hidden, but because we have only ever built tools to find ourselves. This is called the Shadow Biosphere, and it is one of the most quietly unsettling ideas in all of science. The term Shadow Biosphere was introduced in 2006 by Carol Cleland, a philosopher of science at the University of Colorado, and Shelley Copley, a biochemist at the same institution. Their paper argued that we have no scientific basis for concluding that life arose on Earth only once. Abiogenesis, the process by which chemistry becomes biology, is not well understood. We do not know how it happened the first time. If the conditions that produced life 4 billion years ago were common on early Earth, there is no principled reason to assume they produced only one lineage. Cleland and Copley proposed that alternative forms of life could exist today, radically different in their biochemistry, and that we would systematically miss them using every standard method of biological detection. Paul Davies, the physicist and cosmologist at Arizona State University, independently arrived at the same conclusion and became the field's most prominent advocate. In his 2010 book, The Eerie Silence, Davies argued that the search for alien life has been conceptually backwards. We should look for weird life on Earth first, because if we find it, it proves life is not a cosmic accident. If life arose twice on a single planet, it almost certainly arises everywhere. The Shadow Biosphere, in other words, is not just a curiosity. It is a test case for the probability of life in the universe. To understand why shadow life would be invisible, you need to understand what makes all known life the same. Every organism on Earth, every bacterium, every fungus, every whale, every tardigrade, shares the same fundamental molecular architecture. DNA encodes information using 4 nucleobases: adenine, thymine, guanine, and cytosine. That information is read by ribosomes and translated into proteins. Those proteins are made from the same 20 amino acids. It suggests that every living thing on Earth descended from a single common ancestor, what biologists call LUCA, the last universal common ancestor. LUCA is not the origin of life. It is the organism that survived after whatever early competition resolved into a single dominant lineage. The question the Shadow Biosphere poses is, what if some competitors did not die out? What if alternative lineages, built from different chemistry, survived alongside LUCA's descendants, and we simply never noticed? The most striking feature shared by all known life is a property called homochirality. Many molecules can exist in 2 mirror image forms, like a left hand and a right hand, and in a random chemical mixture, both forms appear in roughly equal proportions. But life on Earth uses exclusively left-handed amino acids and exclusively right-handed sugars. No one knows why. This is called the homochirality problem, and it is one of the deepest unsolved mysteries in chemistry. One leading hypothesis is that homochirality arose by chance, a random fluctuation early in life's origin that got locked in and propagated. But if that is true, then a shadow biosphere could have arisen from the opposite chirality. Mirror life, built from D amino acids and L sugars, would be chemically invisible to our enzymes, our antibodies, and our sequencing technology. A mirror bacterium, sitting in a petri dish in your kitchen, could not be detected by any standard microbiological method. In 2024, a team of researchers led by Ting Zhu at the University of Chicago published a landmark study in Science outlining how mirror bacteria might be synthesized and, critically, what the biosafety implications would be. The paper was not a blueprint for building mirror life. It was a warning. Mirror bacteria, the authors argued, would be immune to all known antibiotics, which are chiral molecules that work by binding to left-handed bacterial proteins. Our immune system's antibodies, which are also chiral, would be unable to recognize mirror proteins. A mirror pathogen, if it existed, would be untreatable by any medicine currently in existence. The paper prompted an immediate open letter signed by over 38 prominent scientists, including Nobel laureates, calling for a moratorium on mirror life research. The field had, in trying to understand the shadow biosphere, briefly glimpsed something they did not want to create. The second major framework for alternative biochemistry involves replacing the chemical backbone of life. Standard DNA uses a phosphate sugar backbone, with phosphorus as the key structural element. Phosphorus and arsenic are chemically similar. They sit directly above and below each other on the periodic table, in the same group. In 2010, NASA astrobiologist Felisa Wolf-Simon published a paper in Science claiming that a bacterium called GFAJ1, isolated from California's Mono Lake, could substitute arsenic for phosphorus in its DNA. If true, this would have been the first confirmed example of a fundamentally different biochemistry on Earth. The scientific community responded with intense skepticism. Within 2 years, multiple independent research groups, including a team led by Tobias Erb at the Max Planck Institute, had reanalyzed the samples and concluded that GFAJ1 did not incorporate arsenic into its DNA. The bacterium was simply remarkably arsenic tolerant, using phosphorus obtained from trace contamination in the growth medium. The arsenic life claim collapsed. But the incident illuminated something important. The scientific infrastructure for detecting genuinely alternative biochemistry barely exists. Wolf-Simon's team had used standard detection methods that were never designed to distinguish arsenic DNA from phosphorus DNA at the molecular level. The shadow biosphere, if it exists, would expose this same limitation everywhere. The deepest problem is how we search for microbial life in the first place. Modern microbiology was transformed in the 1980s and 1990s by the advent of culture-independent techniques, most importantly, 16S ribosomal RNA sequencing. Before this, scientists could only study microbes they could grow in a laboratory culture. Since more than 99% of environmental microbes cannot be grown in culture, this meant nearly all of microbial life was invisible. 16S rRNA sequencing changed that by allowing scientists to extract DNA directly from environmental samples, soil, seawater, gut contents, and sequence it without culturing. This revealed an astonishing hidden world. The human gut alone contains an estimated 38 trillion bacteria from thousands of species. But 16S rRNA sequencing has its own deep blind spot. It works by amplifying a specific region of the ribosomal RNA gene that is conserved across all known life. The PCR primers that initiate this amplification are designed to bind to sequences found in standard ribosomal RNA. If an organism uses a fundamentally different ribosomal structure, or no ribosome at all, the primers will not bind. It will simply not be there from the perspective of every tool we are using. Norman Pace, who pioneered environmental DNA sequencing at the University of Colorado, has acknowledged this explicitly. Our detection methods are, by design, searches for life like us. Carl Woese's discovery of the Archaea in 1977 is the most instructive precedent. Before Woese, the tree of life had 2 branches, bacteria and eukaryotes. Everything with a cell nucleus was a eukaryote: plants, animals, fungi, you. Woese, working at the University of Illinois with George Fox, applied ribosomal RNA sequencing to a class of organisms called methanogens, and found that they were genetically distinct from both bacteria and eukaryotes. They were a third domain of life, previously invisible, constituting perhaps 20% of Earth's total biomass. Archaea live in extreme environments, hydrothermal vents, salt flats, the human intestine, and had been systematically misclassified or ignored for decades. Woese faced enormous resistance from the biological establishment. The Nobel laureate Salvador Luria told him directly, "Your Archaea are just bacteria." He was wrong. The discovery of Archaea is a proof of concept. Entire domains of life can hide in plain sight for decades, visible only when someone builds a tool specifically designed to find them. The shadow biosphere hypothesis asks whether we are in the same position today, whether a fourth domain, built from incompatible chemistry, is waiting for someone to build the right tool. Candidates for where shadow life might persist include the deep subsurface biosphere, organisms living kilometers underground on chemical energy, completely isolated from the surface for millions of years, and ancient brine inclusions in salt crystals old enough to predate LUCA's dominance. The acid mine drainage at Iron Mountain in California, pH 0, essentially battery acid, hosts a microbial community so extreme it was initially assumed to be sterile. It was not. Each of these environments represents a place where the biochemical rules of ordinary life are already being bent, and where an alternative biochemistry might have quietly persisted. Researchers have found fragments of DNA in Atacama Desert soil that match no known organism in any global sequence database, with no recognizable homology to any catalog gene from any known domain of life. Most researchers attribute this to sequencing artifacts, or novel organisms whose genes simply haven't been catalogued yet. But some, including Davies, point to these dark sequences as potential shadow life signatures. We do not currently have the tools to rule it out. NASA's astrobiology program has funded research into what it calls agnostic biosignatures, indicators of life that do not assume standard biochemistry. But shadow life might produce none of the chemical signals we watch for, or produce them via reactions we would not recognize as biological. Lee Cronin at the University of Glasgow has developed an approach called assembly theory, which attempts to identify molecules whose structure is so improbable that only a biological process could have produced them, regardless of what chemistry that process uses. Whether it can detect shadow life remains untested. Here is what the Shadow Biosphere forces you to consider. You have been living your entire life surrounded by organisms, trillions of them, on your skin, in your gut, in the air you just breathed. You accept this because science built tools that could find them. What if there are organisms that science has not yet built the tools to find? What if some of them are on your skin right now? What if they are in the soil you walked across this morning? What if the reason we have never confirmed a second origin of life anywhere in the universe is not because life is rare, but because we have only ever looked for one kind? The Shadow Biosphere hypothesis does not claim that shadow life exists. It claims that we cannot currently know whether it does. It means the most fundamental question in biology, how many times has life arisen on this planet, remains genuinely open. The discovery of Archaea required a scientist willing to believe a result that contradicted every assumption in his field. The discovery of the microbiome required abandoning the assumption that only culturable organisms counted. Each expansion of our view of life required first admitting that our previous view was limited. The Shadow Biosphere is the next admission we may be forced to make. And if we make it, if we find, somewhere, the signature of a second genesis, the implications cascade outward in every direction. Life is not a singular miracle. It is what chemistry does when the conditions allow. It happened at least once on this planet. If it happened twice, the question is not whether life exists elsewhere in the universe. The question is whether there is anywhere it does not. Somewhere, right now, something that is neither plant, nor animal, nor bacterium, nor archaeon may be metabolizing. Using chemistry we have not catalogued, with no DNA we can read, and no idea that we exist.